I want to thank the organizers for giving me this chance to speak. The title of my talk is Therapeutic Gene Editing Enabled by New Delivery Vehicles. Cas9-based therapeutics have the potential to revolutionize the treatment of genetic diseases because of their ability to cut DNA with sequence specificity. Cas9-based therapeutics are currently being investigated for the treatment of a wide variety of diseases, such as cystic fibrosis, Duchenne's muscular dystrophy, and sickle cell disease. However, developing Cas9-based therapeutics is challenging because it requires delivering Cas9 protein, guide RNA, and donor DNA in vivo. And this is challenging because Cas9 is a large protein. It also has a guide RNA associated with it that needs to be delivered. And frequently, not only do you have to deliver a Cas9 protein and guide RNA, but you also have to deliver a donor DNA. There is therefore great interest in developing methodologies to deliver Cas9 in vivo, and a lot of progress has been made in this area in the last 10 years. Delivery strategies for Cas9 can be broken down into two broad areas: viral vectors and non-viral vectors. These both have their own pros and cons, which I will talk about on the next few slides. Viral-based Cas9 delivery is by far the most efficient way of delivering Cas9 in vivo, and of the viral-based methods, Cas9 delivery via adeno-associated viruses, or AAV, appears to be the most promising. However, virally delivered Cas9 has several potential limitations. The first one is that if you deliver Cas9 via viral vectors, the Cas9 will be expressed for a very long period of time, at least 4 to 5 years, and this will cause a lot of unwanted non-specific DNA damage because of the continued expression of Cas9 and guide RNA. In addition, the Cas9 is a foreign protein, it's a bacterial protein, and if you continue to express it intracellularly, that will increase the possibility of an MHC1 mediated immune response, leading to the destruction of gene-edited cells. In addition, a large fraction of the human population has pre-existing immunity to AAV and would be ineligible for an AAV-based therapy. And finally, AAV has a fairly small packaging size limit, and if you're trying to accomplish something like homology-directed DNA repair, that requires using 2 AAV delivery vectors, one for the Cas9 and the other for the guide RNA and the donor DNA. And this can only be achieved in animal models if you use extremely high doses of AAV, which are generally far beyond what is clinically acceptable. There is therefore great interest in developing non-viral methods for delivering the Cas9 RNP. Non-viral delivered Cas9 has the benefit that the Cas9 being delivered will be rapidly degraded within 24 hours after delivery, and that avoids the 2 major problems associated with viruses. Because the Cas9 is transient, the amount of off-target DNA damage should get dramatically minimized. In addition, because the Cas9 is transient, the possibility of an MHC1 mediated immune response also pretty much goes away. Cas9 has been delivered non-virally in 3 different formats, either as via a plasmid, mRNA, or as the protein itself, and a variety of nanoparticle-based technologies have been developed to do this. However, in general, non-viral based Cas9 delivery is orders of magnitude less efficient than virally based Cas9 delivery, and there's therefore great interest in developing new types of non-viral delivery strategies for the Cas9 RNP. To address this unmet need, our laboratory has developed a few different strategies for delivering the Cas9 RNP. The first one I'll talk about is a nanoparticle technology called CRISPR-Gold. CRISPR-Gold is schematically shown on this slide here, and CRISPR-Gold is composed of a gold nanoparticle core that has a donor DNA conjugated to it, and adsorbed onto all this is the Cas9 RNP that's schematically shown here. And in addition to having the Cas9 RNP and the donor DNA, CRISPR-Gold also has an endosomal disruptive polymer called PSDET, which has two different functions. It stimulates endocytosis and also disrupt endosomes. So CRISPR-Gold is designed to deliver all the components needed for gene correction via HDR, in particular Cas9, guide RNA, and donor DNA in one nanoparticle. CRISPR-Gold has numerous potential applications. The one we focused on originally was using it for treating a disease called Duchenne's muscular dystrophy. Duchenne's is a genetic disease that has a frequency of about 1 in 3,500 among Caucasian males, so it's a fairly common genetic disease. It only affects males. And there's no cure for this disease. And everybody who gets this disease has a lifespan that only goes up to about the 30s or the 40s. So there's a tremendous unmet medical need in the case of Duchenne's muscular dystrophy. Duchenne's muscular dystrophy is caused by a mutation in the dystrophin protein that's schematically shown here. Dystrophin is a protein that is necessary for muscle tissue to stay healthy. And in patients with DMD, this protein is mutated, and therefore patients with DMD, their muscle tissue starts to waste away, and that eventually causes death by suffocation. Now, Cas9-based therapeutics have the potential to treat Duchenne's muscular dystrophy by cutting the dystrophin gene at the mutation site and correcting it via homology-directed DNA repair. So we therefore investigated if CRISPR-Gold could do this, and we did this in collaboration with Irina Conboy's lab, who's also a professor at UC Berkeley and a muscle biologist. So the experiment we did was we made CRISPR-Gold that had Cas9 protein and guide RNA and donor DNA that was designed to correct the dystrophin mutation, and we injected it into mice and investigated if we could get HDR in the correction of the dystrophin gene. For these experiments, we used the MDX mouse model. The MDX mouse is a transgenic mouse that has a C to T mutation in the dystrophin gene in exon 21, and this generates a stop codon. So MDX mice do not make dystrophin, similar to DMD patients. So we made CRISPR-Gold designed to correct this mutation, injected it into MDX mice in their muscle tissue, isolated the muscle tissue, and then did deep sequencing to see if we could correct the dystrophin gene, the mutated dystrophin gene, back to the wild type sequence. And we analyzed this by deep sequencing. The results are shown here. The y-axis is the HDR efficiency. The x-axis is the various groups. And what you can see is that the mice treated with CRISPR-Gold, their muscle tissue had a 5% conversion rate to the wild type gene. So in the DMD mice, in the MDX mice that were treated with CRISPR-Gold, in the vicinity of the injection, 5.4% of the mutated gene had been corrected back to the wild type sequence. The control, which is the Cas9 RNP, plus donor DNA, had essentially background levels. 5% is actually a reasonably good number. In the case of genetic diseases like Duchenne's, it's predicted that would be around where you would expect to see some clinical benefits. We also analyzed these mice for the re-expression of dystrophin protein via immunohistochemistry. So we injected these mice with CRISPR-Gold, made histology slides, and stained with a dystrophin antibody. The results are shown here. Wild type mice have abundant dystrophin immunostaining. So each one of these little circles here is a muscle fiber that's staining for dystrophin. The control mice, the control MDX mice, which have been treated with nothing, have no evidence of dystrophin, as you can see the blank slide. And the CRISPR-Gold treated mice, CRISPR-Gold treated MDX mice, you see a lot of dystrophin positivity in the vicinity of the injection. And in some cases, it's as high as 60% of the muscle fiber. So we do appear to be inducing the re-expression of the dystrophin protein. One of the benefits of CRISPR-Gold is that it delivers the Cas9 protein itself, which should get rapidly degraded after the editing has occurred. And this should lead to a low amount of off-target DNA damage. So we measured this by doing deep sequencing on MDX mice that had been treated with CRISPR-Gold, where we sequenced 6 sites in the genome that had a very high homology to our guide RNA. The results of this deep sequencing is shown here. And you can see that in general, our off-target DNA damage is extremely low. It's actually below the level of deep sequencing accuracy. So these are all under 1%. The highest one was 0.17%, which is barely at the threshold of being above background. And this is certainly much lower than what you would see with virally delivered Cas9, where generally the off-target DNA damage is in the 1% to 2% level. Finally, we investigated if CRISPR-Gold could improve muscle function in the MDX mouse model. So in this experiment, we treated MDX mice with CRISPR-Gold designed to correct the dystrophin mutation. And then we did a 4-limb hanging test to see how long the mice could hang upside down on a barbed wire mesh. And the results are shown here. The y-axis is the hanging time. The x-axis is the various groups. And what you can see here is that CRISPR-Gold treated mice had a 2-fold increase in hanging time or muscle function over the control mice. They're still below the wild type, but this is certainly a trend in the right direction. So based on these experiments, the lead author of this work was a scientist named Kunwoo Lee. He was a graduate student in my lab. He formed a company based on this project called GenEdit. He's the CEO of GenEdit. And I don't have any equity in GenEdit, so I can collaborate with GenEdit without any problem. And what I'm going to talk about in the next few slides is a collaboration that we've been doing that was done in partnership with GenEdit. So there are numerous organs where gene editing can have therapeutic effects. Of these, the brain is perhaps one of the most exciting. And this is because neurological diseases like Alzheimer's and Huntington's affect millions of people every year. Their incidence is increasing, and there's a great unmet medical need, and new therapeutics are very urgently needed. In addition, developing therapeutics for neurological diseases is inherently challenging because of the formidable barrier of the blood-brain barrier, and also because if you just globally inhibit neuronal signaling pathways, that results in a lot of toxicity. We think that localized gene editing accomplished via an intracranial injection has tremendous therapeutic potential because it overcomes the problem of the blood-brain barrier. And in addition, if you do an intracranial injection of gene editing enzymes, you will only edit the region of the tissue where you inject it, and you will thereby spare the toxic effects of globally inhibiting neuronal signaling pathways. And finally, because gene editing is permanent, you only have to do this once, and intracranial injections are already done in a variety of clinical scenarios. However, despite its potential, very little is known about gene editing in the brain. So we therefore started a collaboration with GenEdit and Professor Hae-Young Lee. Hae-Young is a professor at the University of Texas in San Antonio, and we investigated if CRISPR-Gold could do gene editing in the brain. For these experiments, we used the AI9 mouse model. The AI9 mouse model is shown here, and it's a transgenic mouse that has the TD tomato gene upstream of a stop codon. So AI9 mice do not normally make the red fluorescent protein because of the stop codon. However, if you edit out the stop sequences using CRISPR, you can then get the turn on of red fluorescence. So GenEdit and us, we made CRISPR-Gold that was designed to do this editing. And they figured out how to manufacture and ship it, and they shipped it to the University of Texas in San Antonio. And Hae-Young then injected it into the striatum and hippocampus of AI9 mice, and then looked for red fluorescence. And so the results are shown on the next slide here. So on this slide, every little red dot is a cell that has been cut twice with Cas9 and had a recombination event that led to the elimination of the stop signal, and then the expression of the TD tomato or red fluorescent protein. So you can see in the Cas9 case, this is the mice treated with CRISPR-Gold with Cas9. You can see a lot of little red dots. Each one of those is an edited cell. The control uninjected, you don't see any red. And so this demonstrates that CRISPR-Gold can do gene editing in the brain. For these experiments, we also made CRISPR-Gold complex to CPF1, and Hae-Young injected it into the striatum, and again, she saw a lot of red fluorescence, demonstrating successful editing. Hae-Young was able to get the percentage of cells that had been edited in the brain by doing a simultaneous DAPI staining and then comparing the red versus the blue, so that's what this looks like here. and was then able to get the percentage of cell edited. And what we saw in both cases with CRISPR-Gold and Cas9 and CRISPR-Gold with CPF1 is that we're editing around 10% of the cells. And this is actually a fairly challenging edit to do, because for a cell to be RFP positive, it has to receive 2 cuts of CRISPR and then also recombine. And it turns out the maximum you can get in this animal model is about 25%. So 10% is actually a fairly efficient amount of editing. Based on this, He-Young investigated if CRISPR-Gold could edit neurons. And for these experiments, she used a transgenic mouse model that expresses the YFP gene only in neurons. We made CRISPR-Gold or CRISPR-Gold with either Cas9 or CPF1 that was designed to knock out the YFP gene. She injected it into the hippocampus and then looked for if there was a decrease in the number of YFP positive cells. The results are shown here. The y-axis is the percentage of YFP positive cells, and the x-axis is the control, which is untreated or CRISPR-Gold. And I should mention here that to make a cell YFP negative, all you have to do is cut it once. So this is a much easier type of edit than the AI9 mouse model, where you have to get 2 cuts and also a recombination. So here you just need 1 cut. And so our efficiency is actually much higher here. So wild type is up to about 30% of the cells in a histology slide, whereas if you treat it with CRISPR-Gold, this goes down to about 10%, so almost a 2/3 reduction. And we saw similar things with CPF1. So CRISPR-Gold can do editing of neurons, and it's fairly efficient at knocking out genes. So we therefore investigated if we could have therapeutic effects in treating brain diseases using CRISPR-Gold. The first disease we've looked at is Fragile X-related autism. Fragile X-related autism is the most common hereditary form of autism, and it's a disease caused by a triplet repeat expansion in the FMR1 gene. And this causes the inactivation of the FMR1 gene or the FMR1 protein. The FMR1 protein is an RNA binding protein that acts as a global inhibitor of protein expression. And there's currently no effective treatment for FMR1 related autism, and there's therefore great interest in developing new therapeutic strategies. Now, it's also known that in FMR1 related autism that mGluR5, that the mGluR5 signaling pathway is overactivated. The mGluR5 signaling pathway is shown on this slide here. mGluR5 is an ion channel, and it plays an essential role in learning and memory. And when the mGluR5 signaling pathway gets activated, that results in a large amount of protein expression. And the protein expression is an essential part of mGluR5 signaling. Now, it turns out that in a healthy patient or in a healthy mouse, when you get mGluR5 signaling, the FMR1 protein acts as an inhibitor of mGluR5 signaling, because it will shut down all the proteins that are being expressed due to mGluR5 signaling, and therefore acts as a brake on mGluR5 signaling. However, if you have Fragile X syndrome, which is caused by basically inactivation of the FMR1 protein, so if you have Fragile X syndrome, then you don't have the FMR1 protein, and when you get mGluR5 signaling, that signaling goes undamped and continues to propagate long after it should, because the FMR1 protein is not there to shut down the protein synthesis induced by mGluR5 signaling. So there's therefore great interest in being able to develop inhibitors for mGluR5, and drug companies have spent a lot of time trying to do this. However, this has been challenging because primarily because of toxicity problems. mGluR5 is an essential protein in a variety of other contexts, and if you just globally shut it down, there'll be toxicity. And it also has very similar homology to, and there are also a lot of other ion channels that have homology to it, so it's very hard to selectively knock out mGluR5. In addition, the blood-brain barrier is a challenge. So we therefore investigated if we could use CRISPR-Gold to knock out mGluR5 in FMR1 knockout mice and also in wild-type mice, and rescue mice from Fragile X-related autism. So we made CRISPR-Gold that was designed to do this. We shipped it to He-Young's lab, and she injected it into the striatum and hippocampus and looked for the numbers of mGluR5 negative cells. She did this by immunohistochemistry. And the results are shown here. So the y-axis is the percent of mGluR5 positive cells, and the x-axis is the various groups. And what you can see here is that both in the wild type case and in the FMR1 knockout case, you see about a 50% reduction in the amount of mGluR5 cells after treatment with CRISPR-Gold, demonstrating that CRISPR-Gold can knock out mGluR5 in the brain after intracranial injection. Based on this, we investigated if we could rescue mice from some of the behavioral phenotypes that are caused that are observed in the FMR1 knockout mouse. So it turns out that the FMR1 knockout mouse exhibits many of the repetitive behaviors that are characteristic of autism. One of them is obsessive burying. So if you take the FMR1 knockout mice and put them in a cage with marbles and sand, they will bury all the marbles. And we investigated if knocking out mGluR5 could rescue this phenotype. The results are shown here. The y-axis is the percentage of marbles buried. X-axis is the various groups. So you can see that in the control FMR1 knockout mice, they bury more marbles than the wild type mice, and that the amount of marbles that are buried decreases if you knock out mGluR5 in the striatum using CRISPR-Gold. So here you see a lot of marbles, and here all the marbles are buried. In addition, we also were able to rescue against some of the other phenotypes observed in the FMR1 knockout mice. One of them is the jumping phenotype. So it turns out that FMR1 knockout mice jump up and down obsessively. So that's shown here. This is the amount of jumps in a cage. The y-axis is the jumps. This is the control wild type mice, and this is the control FMR1 knockout mice. You see almost a 20-fold increase in the amount of jumps. However, if you knock out mGluR5 in the striatum with CRISPR-Gold, you get a big reduction in the amount of jumps. I'm now going to switch gears and talk about some other delivery strategies that have been developed in the lab. And the first one is focused on improving the gene editing activity and delivery of CPF1. CPF1 is a fairly recent RNA-guided endonuclease that has emerged as a very exciting alternative or complement to Cas9. And so CPF1, what makes it interesting is that it has a different PAM recognition sequence, which is T-rich, as opposed to Cas9, which has a G-rich PAM sequence. And what this means is that CPF1 can edit regions of the genome that Cas9 cannot. In addition, CPF1 generates sticky ends when it cuts DNA, and that may lead to better HDR and certainly could change the outcome of editing events after DNA repair, and especially in comparison to Cas9, which generates blunt ends. And finally, CPF1 has a 40 base CRRNA, which is much smaller than the 100 base guide RNA of Cas9, so manufacturing the guide RNA in the case of CPF1 will be much easier. Now, in the case of Cas9, it was known that modification and optimization of the guide RNA led to large increases in the gene editing activity of Cas9. However, similar optimizations and chemical modifications of the CPF1 cRNA have not been done yet. We therefore investigated if we could increase the gene editing activity and delivery of CPF1 by engineering its guide RNA. And what we discovered was that if you take the CPF1 cRNA and extend it at the 5' end, you can increase its gene editing activity and delivery into cells in 3 ways. So the first observation was that if you extend the CRRNA at the 5' end, the absolute gene editing rate in cells after electroporation of the RNP is dramatically increased, and this leads to enhanced gene editing. In addition, we can increase the serum stability of the CPF1 cRNA because the cRNA extension is amenable to chemical modifications, which can stabilize it against nucleases, whereas the wild type cRNA is not. And so you can enhance the serum stability because you can now chemically modify the 5' end. And finally, if you just extend the CPF1 cRNA, the negative charge density of the RNP formed after complexing CPF1 is dramatically increased, and that leads to more efficient complexation with cationic delivery vectors and more efficient delivery in vivo. So in this experiment, we investigated if we could increase the gene editing activity of CPF1 by extending the 5' end. So we purchased CPF1 cRNAs that had various extensions ranging from 59 to 4 base extensions. And these cRNAs targeted the GFP gene. We made RNPs with them and electroporated them into GFP hex cells, and then did flow cytometry and got the percentage that were GFP negative. Results are shown here. This is the wild type cRNA, no modification, gives you about 25 to 30% GFP minus, and that dramatically increases almost by twofold. So we're now almost at 60% with the various modifications. So extending it by 4, 9, 15, and 25 all led to dramatic enhancements in gene editing. In addition, we also investigated if these extensions would tolerate chemical modification. So wild type CPF1 does not tolerate modifications of the 5' end, but if you extend it, the extension appears to, and this was demonstrated here, where we took the 4 and 9 base extensions and we modified the extensions with O-methoxy, phosphorothioate, and deoxy, and then looked to see if we still saw an enhancement in the gene editing activity of CPF1. The results are shown here. The y-axis is GFP negative cells, the x-axis is the various groups. So this is the wild type at around 25, and again, the 4 base extension versus the 4 base extension that has methoxy and phosphothioates are about the same. And again, this is again a twofold increase. So we can now do chemical modifications of the CPF 5' end, and we have been able to show that this leads to enhanced stability in serum. And finally, we also investigated if we could use the extensions to enhance delivery. So we made the CPF1 RNP with a 59 base extension, so it's now very negatively charged and complex as cationic polymers. And then we delivered this complex into the muscle tissue of AI9 mice and then looked for editing. So again, AI9 mice turn red when they're gene edited. And so we did an injection in the muscle tissue and analyzed it 6 millimeters away from the injection site. And as you can see from these slides, that there is a lot of gene editing happening up to 6 millimeters away. So we think that the extension will also enhance delivery by making it more negatively charged. In the remaining few minutes, I want to talk about another strategy for delivering the Cas9 RNP, which is focused on making Cas9 RNP peptide conjugates. And we think that Cas9 RNP peptide conjugates have a lot of potential and certainly some benefits over nanoparticle-based technologies, as I presented earlier. And this is because a Cas9 peptide conjugate is going to be much smaller. It's going to be about 10 to 15 nanometers versus nanoparticles, which are generally hundreds to 200s of nanometers. And because of their smaller size, they should be able to diffuse and edit tissues that are going to be inaccessible by nanoparticles. However, a major challenge with making Cas9 RNP peptide conjugates is the fact that if you start chemically modifying the Cas9 RNP, you will most likely kill its nuclease activity. So to overcome this problem, we came up with a linker called TRAILER, which is shown here, which is a disulfide self-amenable linker that allows you to chemically modify the lysine residues of a protein. But then once it goes inside of a cell and sees the glutathione inside of a cell, this glutathione then triggers a self-immolation reaction, which causes the release of the unmodified protein. So the chemical structure of the TRAILER linker is shown here. It's composed of 2 disulfide self-amenable linkers, an ethyl carbonate linker, and a 1,6 elimination linker connected in series. This is the drug and this is the delivery vector. And once you break this bond, you get 2 consecutive self-immolation reactions, which leads to rapid and quantitative release of the amine. And if it's a protein, it's going to be the lysine residue. So we made this reagent here called CPP-TRAILER, which is composed of the cell-penetrating peptide arginine 9 connected to a FITC residue for tracking, and also the TRAILER linker connected to a paranitro carbamate shown here so that you could react it with proteins in one step. So we've made Cas9 conjugated to this TRAILER CPP, and we've investigated what's the release kinetics after incubation in 10 millimolar glutathione. The results are shown here. The y-axis is the Cas9 CPP percent. The x-axis is time. And you can see that in 10 millimolar glutathione, the half-life is about 5 minutes. And this was done by gel electrophoresis, where we're measuring the FITC tag. We've also analyzed the nuclease activity of this Cas9 CPP conjugate before and after reduction. The y-axis is nuclease activity, the x-axis is the various groups. Cas9 CPP by itself has very low nuclease activity, and this is because the CPPs are randomly conjugated to lysine residues and will most likely inhibit the enzyme function because several of the lysines are in the active site. However, if you reduce Cas9 CPP with GSH, you get nuclease activity that's essentially indistinguishable from the wild type Cas9, demonstrating that we can actually release active Cas9 after reduction. Finally, we've investigated if we could do gene editing with Cas9 CPP. So in this experiment, we made Cas9 CPP designed to cut the GFP gene and incubated it with GFP hex cells and then measured the amount of GFP knockout by Sanger sequencing. The results are shown here, and the y-axis is editing efficiency. This is the Cas9, this is the cells that have been treated with Cas9 CPP, and we're at about 5% editing, and it's much higher than the wild type Cas9 added to cells, which is barely at baseline levels. So in conclusion, I've talked about 2 different delivery vectors for transducing the Cas9 RNP in vivo. The first one was called CRISPR-Gold, and the second one was based off of making peptide conjugates with the Cas9 RNP, or CPP-TRAILER. Finally, I'd like to thank the people that did the work. All of the CRISPR-Gold work was done by one very talented graduate student named Kunwu Lee, who's now the CEO of geneti. The CPP-TRAILER work was done by a postdoc named Mao Mao He, who's shown here. And then I also want to thank all of our collaborators who are shown here. And finally, I'd like to thank you for your attention.